Hello dear students, welcome again. In our previous chapter we have learned about inheritance and we have seen the principles of inheritance was explained by the Mendel's experiments. So where Mendel has conducted various experiments and he found that there are some factors which are transferred stably from generation to generation, and later these factors were called as genes. But Mendel was failed to explain about the factors, where are they present and what are they made up of. So these he could not find out, and in order to find out, some of the scientists they tried to conduct the experiments where Walter Sutton and Theodore Boveri they found that this behavior of genes and behavior of chromosomes were similar, and they were thinking that these genes might be present on the chromosomes. So this led to explain the theory of chromosomal theory of inheritance, and then T.H. Morgan was supporting it with his experiment on the fruit flies, where he conducted the experiment on the Drosophila, and he found that there are genes which are associated in the chromosomes, and he called them as this association of genes on the same chromosome is called as the linkage. So the genes which are linked or which are closely associated, they tend to inherit together, so he gave the name linkage. So linkage and recombination, these are the terms explained by the T.H. Morgan, and this strongly supports that genes are present on the chromosomes. So all these experiments of Theodore Boveri, Sutton, T.H. Morgan, they led to support that factors are the genes, or these are the present on the chromosomes. So we know they are present on the chromosomes, but what exactly they are made up of, which molecule is making them, is not to be was not understood for very long. So to understand that, we are going to find out in this chapter on the molecular basis of inheritance. So beginning with the search for the genetic material. So molecular basis of inheritance, in this we already said that Mendel was he has explained the inheritance pattern, and that said that there are factors which are stably passed on from generation to generation, but he did not give a clarity on the nature of factors. Nature of factors means what kind of genes, what are they made up of, the factors, what are they made up of, the genes, what are they made up of, and where are they present, was not given by the Mendel. The investigation began to find out the nature of genetic material, and almost 100 years took 100 years of intensive research to find out that the genetic material is the deoxyribonucleic acid. In short, we call it as DNA. So DNA is belonging to nucleic acids category, and these nucleic acids are ribonucleic acid and deoxyribonucleic acid. So DNA and RNA, these are the nucleic acids, and these are the genetic material for various organisms. But in the most organisms, DNA acts as the genetic material, but whereas in some plant viruses, RNA is acting as the genetic material. But in the eukaryotes and the higher organisms, this RNA is acting as a messenger RNA, which means that this is carrying the message, this is carrying the code from the DNA to the protein. So this messenger RNA role is very interesting, and we'll be understanding this during the protein synthesis. So for now, what we have understood is the DNA and RNA are the nucleic acids, and they are usually acting as genetic material, and in most of the organism DNA is acting as the genetic material, but in some organisms it is RNA, especially in the viruses. But in this chapter, what we are going to understand is we are understanding various concepts. The first one, starting with the search of genetic material. There are several experiments conducted, out of which we are going to discuss very important ones, and these important experiments led to they led to narrow down the genetic material. And then thereafter we study about the structure of DNA. In this we study about Watson-Crick model of DNA, and then we study about DNA replication. DNA has ability to multiply itself, so that is by process called as DNA replication. And DNA, the code on the DNA can be made into RNA by the process called as transcription. So we study transcription, and then we see that genetic code. Genetic code is a nuclear is a code where the coding on the RNA is made into the proteins. So we will understand genetic code. Thereafter, protein synthesis using the RNA sequence, where it is called as translation. And then after the translation, how is this gene expression happening, that also we will discuss. And then Human Genome Project and DNA fingerprinting. So DNA fingerprinting is very interesting because it is used in various investigation projects, like in the maternity disputes or in the crime scenes. They find a proof, and they tend to do this DNA fingerprinting, by which it can be the individual DNA differences can be identified. So these are the various interesting topics under this chapter, and we'll be beginning with the search for the genetic material. The first and foremost experiment, which was simple but it has got the greatest impact, that is transforming principle, explained by Griffith. So it is called as Griffith experiment. So Frederick Griffith, he was a bacteriologist, and in around 1928 he started series of experiments, and he conducted these experiments on the Streptococcus pneumoniae. So this Streptococcus pneumoniae was known to cause the pneumonia. So he has taken mice as his model organism. He has taken this rat or mice as a model organism, and interestingly, this Streptococcus pneumoniae has two strains. So which means that it is same bacteria, but it has two altered forms. So one is R strain, another one is S strain. So R and S are given because of their appearance. R is known for rough strain, S strain is known for smooth strain. So R strain is having a rough appearance on the colonies. On the plate you can see here, these are the colonies present on the bacterial medium. So wherein we see that the colonies of these bacteria are appearing rough compared to the smooth strain. So these rough colonies, because these Streptococcus pneumoniae, they lack the polysaccharide coating around them. They lack that polysaccharide capsule around them. Whereas this S strain, which is smooth strain, which has the smooth appearance on the colonies and their smooth and shiny appearance, this has the polysaccharide capsule around them, which means that this smooth strain is able to secrete the polysaccharides that make them to appear the smooth, to appear smooth. So this gives them the smooth appearance. So these are the R strain and S strain. But what is the advantage S strain has got over the R strain? In the S strain, we see that this polysaccharide coating, which is helping for the protection for this pneumonia, this bacteria, Streptococcus pneumoniae. So this bacteria can escape from the host immunity, which means that even if it is entering into the host, it cannot be it cannot be killed by the host, because it cannot be identified because of its polysaccharide coating around it. So this polysaccharide capsule makes it to escape from the host immunity. Therefore, it escapes and it replicates itself and causes the disease. Therefore, S strain is pathogenic, which means it can kill the organism, whereas R strain is non-pathogenic because it does not have this polysaccharide coating. Therefore, this is subjected to host immunity and then it is killed by the host. So therefore, R strain is killed and by the host. So therefore, R strain cannot be infecting. So therefore, this R strain is non-pathogenic and S strain is pathogenic. So this is a So if you see in a cartoon form, if you see here the R strain, it is having a rough appearance because of lack of polysaccharide coating around it, and whereas this smooth strain, they have a polysaccharide coating around it, and this polysaccharide coating or the capsule makes them to escape from the host immunity. Therefore, S strain is virulent or pathogenic and R strain is avirulent or it is non-pathogenic. So we'll see what is the approach of the Griffith to find out the genetic information, genetic material. So he has conducted this simple experiment, but this simple experiment has got impact, very big impact to find out the genetic material. So here he has taken the R strain, so which means the R strain of Streptococcus pneumoniae is taken and he has injected them into the mice. So here is the mice. The R strain, the rough strain, we said it is non-virulent, right? So this R strain of the bacteria is taken and it is injected into the mouse, and when it is injected, the mouse is able to recognize that this is R strain, and then it is able to defend itself by killing this R strain bacteria, and therefore mouse is living. So R strain, when it is injected, the mouse is living. And coming to S strain, the second experiment is to inject the S strain. The second step is S strain. Griffith has injected the S strain Streptococcus pneumoniae. When he has injected into the mice is not identifying this as a bacteria because of the polysaccharide capsule around the S strain. Because of this, the mouse is not able to identify it, and then mouse is getting the pneumonia, and then therefore it dies. So smooth strain or S strain, it is killing the mouse by the pneumonia. And then here is the interesting part. So Griffith was making a small twist here, that he has taken the S strain. So Griffith killed the bacteria by hitting them. So he has taken the S strain, which is a virulent otherwise, and heat killing means they are subjected to heat, they are boiled or they are given the heat treatment. Thereafter this S strain bacteria were injected into the mice. But when this time S strain, which is heat killed, was injected into the mice is living. This is a surprising result that he has got, that heat killed smooth strain, when he has injected into the mouse was living, which means that this coating, the polysaccharide coating, might be disturbed. Therefore the mouse was able to recognize the bacteria, and therefore it could fight for itself and it was living. But the mouse was living and smooth strain was not able to cause the disease. Therefore on heat killing, this S strain becomes avirulent or non-virulent. So coming to the next very important last step is, when he has mixed heat killed S strain and then live R strain. So he has taken this heat killed S strain, this step, the heat killed S strain, which is non-virulent, and he has taken the R strain. R strain itself is again non-virulent. So when he has taken S strain, which is heat killed, and then R strain, which is already non-virulent, so when he has mixed both of these, so S strain, which is heat killed, and then R strain, which is living, so when he has mixed both of these, and when he has injected into the mice, he finds that the mouse is dead. So the mouse is dead because this rough strain and heat killed S strain, when they are mixed together, the mouse is dead. So this is leading to very important conclusion, that there is something which is transforming the R strain into the S strain. So in order to find that, what he has done is, after the mouse is dead, he took the injection, he tried to extract back what kind of bacteria is present inside. To his surprise, when he has extracted the blood sample from the mouse, from the heart, he found that it had S strain bacteria, which are living ones. So it means that the S strain has some genetic material which is transforming the R strain into the S strain. Therefore we call this as transforming principle. So this is again a depiction how we can see that this is smooth strain, which is virulent, and this has polysaccharide coating. When it is injected, the mouse dies. And rough strain, which is non-virulent, when it is injected, the mouse lives. And then heat killed smooth strain, S strain, these are losing the polysaccharide coating, and when they are injected, the mouse lives. And but when these two non-virulent heat killed S strain and then rough R strain, when they are injected together, the mouse dies surprisingly. This led to the conclusion that R strain bacteria somehow were transformed by the heat killed S strain bacteria. So there means there is a transformation of R strain into the S strain, therefore it becomes virulent, and something in the heat killed S strain were transferred previously harmless R strain into the making the R strain deadly again. So this must be due to the transfer of the genetic material. That is what he said. So there is involvement of genetic material which is transferring the character of virulence from R strain to the S strain. So but he could not define the biochemical nature of this genetic material. So the Griffith was not able to give the biochemical nature, which means he could not say what are these genetic material made up of. So what exactly is this genetic material was not given by the Griffith. But this experiment has given a greater impact, and even after around 16 years after that experiment of Griffith, it led these scientists, Avery, McCarty and McLeod. These scientists tried to conduct the same experiment as the Griffith conducted. So the same Streptococcus pneumoniae, R strain and S strain, they have taken the mouse and they tried to replicate the same kind of experiment again. So that is what we study under this experiment, that biochemical characterization of transforming principle, which means that these three scientists are explaining what is the biochemistry or what are the biomolecules involved under the genetic material or that transforming principle. So here we are going to see the very simple experiment, and in this experiment what they have done is, prior to these people conducting the transforming principle, before these people trying to do the biochemical characterization of this, earlier it was thought that genetic material was thought to be the protein, which is a wrong notion, which is proved by these Avery, McLeod and McCarty. These have proved something else. We are going to see that. So they tried to determine the biochemical nature of this Griffith transforming principle, which means that they have conducted the same Griffith transforming principle experiment, and they saw what is the molecule present in that. So to identify, they had a different approach. So what they have done is, they have purified biochemicals. They have taken the blood sample and they have taken all the molecules present in it, both proteins, DNA, RNA and other kind of biomolecules. So all these biochemical molecules are taken from the heat killed S strain. So important here is heat killed S strain, and then these are tested for the transformation with each biochemical experiment. We are going to understand here. So first what they have done is, they have taken this heat killed S strain. So here this picture is a better depiction of this experiment. So they want to determine what is the genetic material here. So what they have done is, they have taken heat killed S cells or heat killed S strain bacteria, and they have already extracted the biomolecules from that heat killed S strain. So they have taken all the biomolecules here, and they tried to split into various ways. And the first way is, they are taking the entire sample apart from the protein. So first they want to see that if there is no protein, they want to remove the protein from that biomolecules. So if there is no protein, then what would happen if the other biomolecules, whether they are able to transform the bacteria into the S strain, is what we have to see here. So they have taken the biomolecules first from the heat killed S cells, and they have split these into various media, and first one they have added is proteases. So when they have added these proteinase or proteases, these proteases, what they do is, they digest only the protein, which means that in these biomolecules, of all the biomolecules, protein is not here, which means that we will understand whether the protein, if the protein's role is there in the transformation or not. So they want to remove it first. So they removed protein first. So they added the proteases, which are the digestive enzymes to proteins. So proteins are digested by the proteinases. So when they have added proteinases here, the protein is digested, which means that all the biomolecules are present except the protein. So they have tested here and they have added the R cells here to these heat killed S strain molecules here. So when they have added the R cells have transformed into the S cells here, which means that the protein was not involved in the transformation. Even without protein, there is a transformation occurring here. We see that the R cells, when they are added to these molecules, the rest of the molecules, they have made this R strain into the S strain, right? So therefore there is transformation occurring even in absence of the protein, which means that protein is not the genetic material. Next one is they have taken ribonucleases, that is RNases. RNases removes the RNA. So they have taken the same strain biomolecules here. From that biomolecules, they want to remove one important molecule, that is RNA. So in order to remove the RNA, what they have done is they have taken RNA digestive enzymes. The RNA digestive enzymes are nothing but ribonucleases. So these ribonucleases they are added to this medium, this biochemical. So therefore it digests only RNA, which means that except RNA, all other biomolecules are actively present here. So in this case, what we see is when they are added with the R cells in these biomolecules, they see that R cells are also getting transformed into the S cells, which means that the transformation is still occurring even in absence of RNA, which means the RNA is not the genetic material here. So RNA cannot be the genetic material. Therefore, so far we have seen proteases, when they are added, protein could not affect the transformation, and RNases also could not affect the transformation. Even in absence of protein and RNA, the transformation is still occurring, which means that there is another molecule which is responsible for this transformation. And this time the third sample, what they have done is they have taken DNases, that is deoxyribonucleases. So these deoxyribonucleases, what they do is they digest the DNA, or they take away the DNA, or they break the DNA. So these DNases, when they are added to this sample, which means that this sample does not have DNA. So apart from the DNA, all other biomolecules are present, like proteins, RNA, all the other molecules are present here except DNA. So when they have done this, when they have taken this sample and when they have allowed the R cells, allowed the R strain bacteria into this R strain bacteria was not able to transform. So there was no transformation seen here. Therefore we say that without DNA there is no transformation, which means that this molecule of DNA is responsible for the transformation. So this narrows down to a point that DNA is the genetic material. So this is very important experiment to prove that DNA is the genetic material, because they have taken DNases here. DNases digest the DNA, and in absence of DNA, this smooth strain, the S cell molecules, could not make the R strain molecules into the S strain. So which means there is no transformation without DNA, which means DNA is the genetic material. So here purified biochemicals, both proteins, RNA, DNA, from the heat killed S strain molecules, they are tested for the transformation. So here protein digestive enzyme, that is proteases, and then RNA digestive enzyme, that is RNases, so they could not affect the transformation. But the DNA digestive enzymes, that are DNases, they inhibited the transformation. So this suggests that DNA was responsible for the transformation, and this experiment leads to conclusion that DNA is the genetic material. So very important experiment which led to conclusion of DNA is the experiment. Through this we have found out. So these are the two important experiments we discuss in this class, and then we'll move on to one another experiment which is very important again to prove that DNA is the genetic material. Okay, so thank you. If you have any doubts, we will inquire it here.